Good morning, everyone. I'm Kevin. I'm a research scientist in the Matson lab at Fred Hutch, and it's my pleasure to talk about some work we've published recently, nucleotide context models, outperform protein language models for predicting antibody affinity maturation. Okay, so our lab is interested in computational models of antibodies. So antibodies are essential to the adaptive immune response, and computational models have been used to inform antibody engineering and vaccine development. There's lots of B-cell receptor sequence data available now, and researchers are rapidly accumulating more. So as a result, there's been many new models that have emerged in recent years, in particular protein language models. So these are models that are using the machine learning techniques found in large language models like ChatGPT, but they're trained on antibody sequences instead of text. And so a natural question is, how do all these various modeling approaches compare? And so that brings me to sort of the purpose of our study, which was evaluating performance of model predictions over the course of affinity maturation. So first, I should maybe explain what I mean by over the course of affinity maturation. So affinity maturation is the process in which our immune systems generate potent and specific antibodies through B solution. So here's a cartoon. So at the top here, B cells compete to bind antigen with the receptors, and those that are able to capture receptors are selected by interacting with T cell helper T cells, and selected B cells undergo division, somatic hypermutation, which then can create clonal mutants that have better receptors, which will then go on to outcompete other B cells. And so this sort of clonal development can be described with a phylogenetic tree. And so, the naive V cell that started the lineage is at the root at the top of the tree. And then, so each level of the tree corresponds to cycles of mutation and selection. And then the final mature B cells that emerge are at the bottom, at the leaves of the tree. Now, practically speaking, most of the time, what we observe experimentally are just the mature B cells, so just the leaves. And so we have to computationally infer the naive sequence, the tree structure, and the ancestral sequences. So now, going back to the point, what we mean by over the course of affinity maturation, what we want to do is analyze the B-cell evolution, step by step or branch by branch of these trees. And this is following work by Spizak and others on their work on modeling somatic hypermutation. And so basically what we're doing is turning our data set of BCR sequences into a collection of trees and then a collection of branches where each branch corresponds to a parent sequence and a child sequence. And to do this is important because mutation effects, mutation patterns, and selection pressures depend on the sequence. So as the sequence changes along the lineage, so do those patterns. Okay. So next, I want to talk about what we mean by model predictions. So let's look at a single branch or a parent-child pair. And what we want to do is have a model predict possible outcomes based on the parent sequence and then be able to eventually compare it with the child sequence to see how well the model does. So obviously the input to these models is the parent sequence, but we also need to account for the amount of evolutionary time between parent and child. So that's also a factor into the model. And what we do with the output of the model is compute the probabilities for the amino acid outcomes for all the sites. So the prediction is a matrix where each row corresponds to a amino acid site in the sequence, and each row has 20 probabilities for the 20 amino acids. Now, an example of a model that we evaluated was the S5F model, which we took from the Shazam repository. And so this is the classic model for somatic hypermutation, where it reports a mutation rate for every nucleotide 5-mer context. And so this is a model that takes in only the nucleotide context. Now, another model that we looked at, a language model example, is the AbLang2 model. This is a language model trained on antibody sequences. In principle, it should be predicting mutation and selection effects. It was trained on amino acid sequences. So that means that it only looks at the amino acid sequence instead of the nucleotide sequence. But beyond the fact that, S5F takes in nucleotide inputs and AbLang2 takes in amino acid inputs, these two models are also representative of what we consider sort of two contrasts in perspective on how to model affinity maturation. So S5F falls under what we call the molecular evolution perspective, which means this is the sort of the more traditional approach where we know that certain patterns of affinity maturation, like somatic hypermutation, depends on certain properties of DNA. And so we build our models on explicitly modeling these features with the nucleotide sequence context. Now, in contrast, the language modeling approach is more implicit. The assumption here is that protein properties are encoded in amino acid sequences. So you train these models on lots and lots of amino acid sequences. And the hope is that properties like binding affinity will correlate with what the model reports as the probability for that sequence. So as I mentioned earlier, S5F is an example of a molecular evolution model. AbLang2 is an example of a language model. Another example of a molecular evolution model that we evaluated is Thrifty, which comes from our lab. This is a model that was initially designed to model somatic hypermutation, was trained on non-functional sequences. It uses a machine learning approach using a convolution as a way to effectively look at a 13-mer window. But we also took this model framework and trained it on functional sequences to get a thrifty prod model, which has both selection and mutation effects. On the other hand, for language modeling, another language model we looked at was ESM-1v. This is a model that's trained on just general protein sequences, but it has been used in studies for antibody engineering. So we decided it was worthwhile to compare in our studies as well. Okay, so we also made an attempt to combine models from both the molecular evolution approach and the language modeling approach. And essentially what we're doing is taking the probabilities coming from a mutation model like S5F or Thrifty SHM, and then sort of multiply it with a selection factor that would compete with a language model, in this case, ESM. So a selection factor is a number. If it's greater than 1, then it means that the potential amino acid outcome is more preferred than the amino acid and parent. And if it's less than 1, then it's not preferred over the parent. Okay. So yeah, let's get to some results. Evaluating performance. So to evaluate performance, we need to find some way to compare, observed some observations with predictions, and also have some metrics to quantify, these comparisons. So yeah, let's go back to, again, a single branch, parent-child pair. And so what we're going to call an observed amino acid substitution is just differences between parent and the child. And now this is for one branch. We can say tally up all the substitutions across all the branches in our data set according to sequence position. And we could generate a distribution like this, which is just the number of amino acid substitutions at each site where the site is aligned to the IMGT position. Here, this is a distribution generated from a data set that has 440 parent-child pairs. For those that have seen these distributions of mutations before, you can probably guess that these peaks here correspond to the CDR regions, which are the regions of the antibody that come into contact with the antigen. I just want to point out, this weird dip here in the CDR3. This is just an artifact of using the IMGT numbering and how they introduce numbers for different CDR3 lengths, and the fact that we're aligning sequences with different CDR3 lengths. So there's nothing real or biological here. Now, so this is the observed distribution. If we pick a model, we can also generate a expected distribution. So what we do here is, again, the model predicts probability for each amino acid outcome at every site. So we can just sum up those probabilities for amino acid change over all the branches. And now in the bar plot, we get what is the expected number of substitutions by the model. In this case, this is the model for S5F. And the blue bars correspond to sites that are in the framework region. The yellow bars are the sites in the CDR region. And so now we need to quantify, how well is this agreement. So the metric we come up with is what we call overlap. Basically, it's just the ratio of the area of the intersection of the two distributions divided by the average of the two areas. And so for S5F, with its data set, we get an overlap of 0.783. And so how does this compare with other models? Well, let's look at how S5F compares with AbLang2. So actually, it doesn't do worse than AbLang2, which is interesting because S5F is just a model for somatic hypermutation, while AbLang2 should model both effects, selection and mutation. I think this points to the fact that, select in terms of predicting where amino acid substitutions happen, somatic hypermutation is just a much more important and dominant effect. But also it suggests that the nucleotide context is important to have a very precise modeling of somatic hypermutation, which AbLang2 lacks because it's only looking at amino acid sequences. Now I'll show one more other distribution. This is the thrifty prod model on top. So again, a reminder, this is a molecular evolution model, but it's trained on functional sequences. So it is also has selection and mutation effects. It has a much better overlap of 0.923, and it turns out this thrifty prod model was the best performing model overall in our study. So just to kind of summarize, this overlap metric for all the models here in sort of a tabular form. So the first three models listed are models that are under the molecular evolution perspective. AbLang2 and ESM are the language models, and then below are the attempts at combining the two approaches. And so, yeah, the molecular evolution models are doing better than the language models. Interestingly, the combined models do much worse. We can also evaluate these models in each region, each framework and CDR region separately. And the only point I want to make here is that if you look at thrifty prod, it is better than all the other models across all regions. So this overlap metric is allowing us to sort of evaluate how models predict where mutations, amino acid substitutions happen. We would also like to understand how well models predict, the exact amino acid outcome. And for this, we compute a quantity called perplexity. The only takeaway I think I want you to get from this equation is that this is 1 over the model probability. And so what that means is the probability is high, this perplexity quantity is going to be low, and that's a good thing. If these probabilities are small, then the perplexity is going to be high, and that's not great for the model. And so the probability we're computing here is the probability for the amino acid that is observed in the child. So for example, in this parent-child example, this P would be the probability to get an alanine at this site. And we evaluate, this is a running product, so we evaluate this over all the substitutions that happen in the data set. And then this 1 over n just means that ultimately the perplexity quantity is a geometric mean. And so if a particular model has a lot of high probabilities for all the substitutions, then this means that the model is generally confident and correct, and we have a low perplexity score. Whereas if it reports a lot of low probabilities, then it seems to be very perplexed by the observed substitutions, and it will get a high perplexity score. Okay, so there's not really an interesting graphic for perplexity, so I'll just jump right to the perplexity summary. And so, yeah, once again, here are the list of models and the overall perplexity scores. Again, lower is better, or, in the color scheme case, darker colors is better. And again, for the most part, the molecular evolution models do better than the language models. So certainly all three does better than ESM, while AbLang manages to beat out S5F here. And once again, the combined models don't do very well. Now, evaluated in each framework and CDR region, there is sort of an overall pattern where for each model, we get better perplexity scores in the framework regions compared to the CDR regions. And we think this is just reflection of the fact that there are more sites under purifying selection in the framework region. So this makes for a, say, easier prediction task to get correct. So yeah, I mean, I've mentioned so far that the combined models of molecular evolution and language models don't do so well. But before concluding, I would like to talk about some follow-up work we've done where we have a more promising model that does combine the two approaches. And so once again, we get, this idea of having a mutation multiplied with selection. Once again, we're going to use a mutation model that's already available. In this case, Thrifty SHM. But what we're going to do now is use a language model to train for the selection factor. Essentially, the idea here is we're going to have a mutation model subtract off the mutation effects and train a model to learn the residual effects, which we'll interpret as being due to selection. And so we call this model the deep amino acid substitution model, the DASM model. And here is a distribution of observed versus predicted, again, aligned in IMGT positions. Now this is the performance of a mutation model Thrifty with a DASM model providing the selection factors, and we get a very high overlap. Again, you can see this visually. Now this is maybe not all too surprising because we're training this to basically model affinity maturation, and so this is kind of expected. But what is interesting is that we find that the DASM selection factors correlate better with functional measurements. So I'm showing a table here where we're comparing DASM with different protein or antibody language models. And what we're doing here is computing the correlation values with binding and expression measurements with respect to a deep mutational scan of an antibody. And so you can see here the correlation is higher for DASM compared to the other models. And so this kind of gives us a lot of encouragement that this is showing very promising in terms of interpretability, and that it's actually capturing some real biological effects. So yeah, to conclude, we found that nuclear context is crucial to accurately model antibodies and affinity maturation. And as a follow-up, we've developed this DASM model, which merges the molecular evolution and language model approaches, and it looks very promising. And so lastly, I just thank members of the Batson Lab, our collaborators, and those that have shared their data with us, and also Fred Hutch, HHMI, and NIH. And I'm happy to take questions.